Yeah.
All right.
As Rashid said, my name is Erich Miller. I work with Dr. Erich Miller and Dr. Joseph Kramer at Alvernia University, and my research is specialized around neuroscience and the study of complex neuropsychiatric conditions. We try and study the treatments used to treat things like schizophrenia, bipolar disorder, or OCD, and try and find better treatments and also try and find the neurological basis of the conditions. And with Joseph Kramer, my specialty is in inorganic chemical synthesis, such as ruthenium coordination complexes and, to some degree, cobalt complexes. But today, the majority of the presentation will be about what I'm doing right now with Dr. Rectemalt, because I'm being paid by Alvernia to do my research on, we're currently analyzing we did a biochemical analysis of 6 medications used to treat obsessive compulsive disorder, and now we're looking, are psychiatrists actually prescribing that? So what turned from a basic neuroscience project I designed probably the latter part of high school has now turned into an international epidemiological analysis.
And I can confirm from him that he was talking about this with me during high school. I remember in Anaptys 1 and 2, he kept saying, you know, I'm really interested in this. I really want to do this. And then you just ended up doing it.
Yep.
So, which is pretty cool.
Okay. So, I always like to give a good amount of background before I just discuss research. So we really need to look at first what is OCD. And this is the condition I decided to specialize in for a number of reasons. And probably a lot of individuals we've heard about OCD, but a lot of the things we hear in the actual media is not what OCD is. It isn't a finicky miss to have symmetry and whatnot. Actually, what OCD describes is it's a condition in which a person's thoughts become looped, essentially. They become obsessed in a certain cycle. Someone could be obsessed about the same topic for 3 days at a time. And the only thing to break that cycle is to perform a compulsive behavior, which a compulsion could be a number of things. We've all heard this typical hand washing and the other things, but there are a number of interesting cases. I actually had just read a case study of someone who knew that they could not contract type 1 diabetes from another person and yet believed that they could and continue to believe that, and that's exactly what we deal with typically when we're doing OCD patients. The relative prevalence is not a very common neuropsychiatric condition. Actually, only about 1% of the global population is afflicted with OCD. That comes out to about, in America, around 3 to 4 million individuals out of the entire United States. A bit of history, these are just 2 individuals that were very renowned for having OCD. It's Nikola Tesla and Howard Hughes, who's quite renowned for it. Actually, they made a movie on it, The Aviator. The nature of OCD. So one part that we have to accomplish when we're doing a biochemical analysis is we need to look at the factors that underlie the condition. And OCD is primarily a genetic precedented disorder. There is epigenetic factors involved, but primarily we look at the genetics that might cause it, and there's actually currently an international collaborative going on to find the genetics that predispose for its development. But one gene I know off the top of my head, which is listed here, is this on chromosome 17. One of these select regions binds for a gene known as the SLC6A4 protein, or gene which encodes for the serotonin transport protein, also known as SERT, which I'll get into later on. SERT is our main target when we look at SSRIs from a pharmaceutical chemistry standpoint, because that controls the regulation of serotonin within the synapse. And this is the actual gene that encodes for that. OCD is believed to be predisposed by 5-HT abnormalities, which 5-HT is the scientific name for serotonin, 5-hydroxytryptamine. Serotonin abnormalities and glutamate abnormalities underlie the condition. Specific brain regions that this occurs in is the caudate nucleus, the cortical striatal thalamic cortical regions, which are towards the more towards the posterior end of the head, posterior and down. But majority of the condition is believed to be genetic and to some degree epigenetic. Some factors, environmental factors, are believed to trigger it. High stress environments are believed that they can trigger the genes into activation. But the consensus among the community is that there is some sort of genetic precedence for the condition, much like many other neuropsychiatric illnesses. So treatments of OCD, and this is where the research begins to come in. There are only 2 approved first-line treatments for OCD. There's CBT with ERP, which stands for Cognitive Behavioral Therapy and Environment Exposure Response Prevention, and SSRIs. My specialty is not more in the psychological aspect, so we focus on the SSRI medications because of the chemistry. The second line is clomipramine, which is a tricyclic. We don't really associate with that much anymore because tricyclic antidepressants are kind of being pushed out a bit. And antipsychotic intervention, which we don't cover because that's an entirely separate neurotransmission system that primarily targets dopamine, and that's only given to people that there's little effectiveness. So this brings us to the science of it, the chemistry. SSRIs and 5-HT dysfunction. So SSRIs are relevant to OCD treatment because they target a protein, as I said before, known as SIRT. SIRT, by targeting it and binding to it, allows for extra serotonin to build up in the synapse because it prevents the reuptake into the presynaptic neuron. These drugs bind in a number of different ways, and we'll get to the relevancy of why they're different, because otherwise these medications here, being the SSRIs, all do the same function, primary function, and that's inhibit the SIRT protein from reuptaking serotonin. However, they have non-selective binding abilities that we'll cover then that give them their special properties. SSRIs, the exact biochemical method of which they bring therapeutic responses in individuals is not known perfectly. We believe it has to do with the fact that it increases binding to select 5-HT receptors, which increases a therapeutic response, such as 5-HT1A or 5-HT2C, both receptors that are hypothesized to be linked to anxiolytic properties, which means they reduce anxiety. But there's also a theory that they increase neuroproliferation and neurogeneration, which means that they actually assist in forming new synapses between other neurons. So our 6 SSRIs, and these are their generic names, they have quite complex IUPAC names, but we have fluvoxamine, sinister citalopram, which is what the S stands for, racemic citalopram, which is RS, sertraline, fluoxetine, and paroxetine, which the brand names of these medications in that order are Luvox, escitalopram, Celexa, Zoloft, Prozac, and Paxil. And this diagram here shows the actual method through which serotonin in the synapse, tryptophan within the synapse becomes serotonin. We have our L-tryptophan molecule that undergoes hydroxylation by tryptophan hydroxylase, and then decarboxylation by dopa decarboxylase, which forms our serotonin molecule. And dopa decarboxylase is an enzyme that is the last step between serotonin and tryptophan, that's actually 5-hydroxytryptophan, that middle molecule. So these are the actual structures of our 6 drugs, and if you guys haven't been versed yet in line angle, this might look like a bit of a mess at the moment, but these are the actual structures, and this is what we analyze in order to see like the biochemical properties. Now, when you look at these molecules, they all have one striking similarity. It might be hard to see because the molecules are a bit small, but each molecule has a halogen group on the actual molecule. Here we have chlorine, fluorine, fluorine. Most of them are fluorine. You do have a chlorine and sertraline. This is how the drug actually binds to SIRT. It binds through a function known as van der Waals forces of attraction and binds to the halogen atoms. For example, paroxetine binds with the greatest potency because it has the strongest yield to the orthosteric binding site of the SIRT protein, which the orthosteric site is the active site, the primary active site. We talk about allosteric sites too, but that's usually more relevant when we're looking at the citalopram group. And interestingly, we can't quite figure out why citalopram has a nitrile group on the end of it, because typically nitrile groups are not favored by the body because they break down into a cyanide complex. But overall, these are the actual structures of the drug. And when you see these little letters here, this is chirality. And if there's one thing I can't stress enough when it comes to pharmaceutical chemistry, chirality is everything. Chirality refers to the direction that the actual molecule has. So we have 2 directions in molecules. We have R and we have S, which is rectus and sinister. Chirality actually, molecules can be optically active, which means they reflect light and bend in certain directions. But chirality is really important when we talk about medications because I can give you a great example with thalidomide. Thalidomide was sold as a racemic structure, R and S. The S portion of thalidomide was the portion that actually could have been capable of stopping morning sickness, but it was R thalidomide that caused mitotic spindle fiber deformation, which ultimately caused mutations within children in vivo. SSRI pharmacodynamics. So this is more what I specialize in. This is pharmacodynamics is the study of how the drugs actually bind within the brain. It's where they go, what they do. Also outside the brain within the PNS, but majority of our focus is on the CNS because the PNS is more focused on muscarinic receptors, histaminergic, stuff like that. But the SSRIs are very similar in that they all bind primarily to the orthosteric site, but they all have secondary sites. Acetyloprene, for example, is the highest selective medication for just the SERT transporter protein. It binds orthosterically and allosterically to the protein, and then the next closest affinity site we have is 5-HT2C, which is just a serotonin receptor on the postsynaptic neuron. Whereas with Prozac, fluoxetine, that is abundant. That binds non-selectively to virtually every 5-HT receptor, which can elicit a number of responses depending on whether it's an antagonist, an agonist, an inverse agonist, which means it actually does the opposite of what a normal agonist does, but it produces the same response. And that's ultimately what defines the differences between these medications, is where they bind non-selectively, because they all target the same thing, which is why they're all classed as selective serotonin reuptake inhibitors. Otherwise, they would fit into different classes. Sertraline has been debated as a potential one that also could be considered a norepinephrine reuptake inhibitor, but that's never been changed. And then the biggest differences lie in pharmacokinetics, which pharmacokinetics is a fascinating field that studies how the drugs break down. And this is exponentially relevant when you're prescribing medication, because if you don't know the pharmacokinetics of a specific person, you could kill them by accidentally prescribing medication that counteracts. So, for example, before I get into that, I'd like to show an example of the actual process in which an SSRI would exude its response. So here we have a normal serotonin synapse, and up here is the actual shorthand version of tryptophan to serotonin. But serotonin is released through vesicles from the presynaptic neuron and goes into the synaptic cleft. Now, normally this would be abundant with receptors, except in individuals with neuropsychiatric conditions. People with major depression, OCD, and schizophrenia are believed to have a far lower number of receptors than other normal, healthy individuals. However, once serotonin gets into this cleft, this protein here is meant to act as the 5-HTR, which is also an interesting way. Well, this actually is a 5-HT receptor, but this here is our SERT protein. This is our target site for SSRIs. Typically they bind, it's like it's on the side, the binding site we believe, but typically they bind here, and thus this prevents serotonin from being reuptake, and it continues to build in here. And this we'll get into shortly with pharmacokinetics. And MAO stands for a monoamine oxidase, which is primarily used to break down serotonin in the body. And there are medications that also target that, known as oxidase inhibitors. And this here is just a diagram. There are sodium-dependent channels, ligand voltage-gated ion channels within the postsynaptic membrane also, but those are mostly used to maintain ionic stability. Pharmacokinetics. Now this was when I first encountered this field, this was gibberish. When I encountered this field first time, I had to have assistance from an organic chem professor to learn this. Pharmacokinetics is the study of how hepatic enzymes and various other enzymes we have in our body break down these medications and then excrete them and various other factors. So why do pharmacokinetics matter? Well, your body has about 80 enzymes that 80 hepatic enzymes that it uses to break down most major medications. But there are 5 that break down almost all medications. And can't name all of them off the top of my head, but a few of them, the main ones are CYP2D6, 2C19, 3A4, 3A5, and 1A2. I think I did name them all.
We believe it.
So these are the main enzymes that are used to break down the medications, but CYP2D6 is the one that has the major abundance, or the major function in breaking down SSRIs. So CYP2D6, let's say you take a medication that it targets that, and it needs that to break down. But if you take another medication that also requires CYP2D6, you will get what is known as either increased efficacy measures or neurotoxicity. Because the problem is the drugs will compete for the enzymes. And eventually one drug will then be left out, so to speak, and the bioavailability will increase. And that can be good if you're trying to make certain drugs increase in bioavailability and make them more potent and powerful, last longer, it increases the half-life, but It can also kill someone if you're not careful, depending on the combination, or it can severely injure them. And that half-life really comes into everything, and that's one thing a lot of individuals I find. I love half-life when we talk about it in chemistry because it really shows how medications work. So, for example, citalopram has a half-life around 24 hours, which means that the medication, that's about the time it takes for the medication to be excreted about halfway out of the system, which is different from steady state concentration, which refers to when the concentration intake in the body and also out of the body are equal. But fluoxetine, for example, has a half-life of 2 to 4 days, which means that it doesn't, the protein or the chemical itself is not excreted for 2 to 4 days after one intake. Unless you, of course, have polymorphisms, which polymorphisms refer to genetic variations of the protein, which are very abundant in CYP2D6, which is actually, as far as we know, the most polymorphic enzyme that is out right now. It virtually changes. Everybody could potentially have a different version of CYP2D6. MAO stands for monoamine oxidase. These are the enzymes that primarily break down serotonin as well as other, they are also present in other synapses and break down other neurotransmitters. And we have catechol O-methyltransferase, which is used to, actually this can directly break down certain medications. Paroxetine is the main medication that gets broken down by COMT, and it forms different catecholized products. So then this brings us to the actual design. My project initially began with one medication. We started with fluvoxamine, we did a biochemical analysis on it, and then eventually, as I networked, we evolved it to 6 medications, and we did a biochemical analysis on all 6. But then it occurred to us that we need to see if the project actually transitioned from a neuroscience experiment then into an epidemiological experiment, and now we're analyzing, are psychiatrists actually prescribing these medications that are designed best for specific conditions? And we're getting interesting responses. So we currently measure the SSRI prescription frequency versus reported efficacy. Paroxetine had too low of a number to be significant at the moment, and the project is still ongoing, these are preliminary figures. But sertraline, which is the largest medication we have in terms of company that makes it, being Pfizer, is the most selective medication, but was rated with only 70% efficacy as compared to fluoxetine, which had up to 87% efficacy and was made by a smaller company. Now, fluoxetine given isn't the oldest SSRI. It is the oldest one. So we're beginning to find that individuals who chose this also reported that the reason they chose it is because of long-term reliability. However, the newest drug, citalopram, has already come in. Now, it's not as high a number in terms of response rate, but it's already coming in with higher efficacy than sertraline, and we're beginning to find that there seems to be monetary things involved with certain prescription trends. Bigger companies' drugs are getting marketed more than smaller companies. And these are this is a loaded graph. These are the number of reasons that people have given as to why they chose a specific medication. So our number 2 choices are validated efficacy in clinical trials and professional experience. Professional experience typically encompasses multiple factors. It's essentially saying that the person feels that they are confident in their choice. But validated efficacy, the individuals who chose sertraline chose validated efficacy as the reason behind why they chose the medication, and yet the efficacy seems to have faltered compared to other medications that are far more beneficial, far more effective. And actually, in terms of pharmacodynamic and biochemical analysis, the most effective drug should be citalopram in terms of its capability to modulate serotonin. And these are the number of reasons. Minimal adverse effects came in. We had a few people who took into effect pharmacogenetic considerations. That's a new field where they actually will sample test your enzymes in order to see if you can actually lack specific enzymes and therefore can't metabolize drugs properly, or you can have polymorphic versions that are high metabolizers and they break the drug down way too quickly. Essentially, this is just a graph illustrating the average response rate versus the rationale they gave for why they made their decision. The study reception. For the most part, the study has come back with decent data so far, and individuals have, the feedback from individuals is pretty good and constructive. We did have one negative person that did attempt, we learned how, we had to defend our research against one person who was attempting to discredit it with information, and he didn't back his point with any facts, so that was something me and Rectum had to deal with. But for the most part, the study reception has been very good. And project goals. Desired outcome, we've been writing a paper on this entire analysis for over 1.5 years. The paper is 30 pages long and is set to be published in the International Journal of Obsessive Compulsive Disorders. We're preparing it to be put in press. We're hoping it gets accepted. It's very long editing a paper. It's a very long process. It's on its 5th major draft, and that's the draft that, in terms of minor edits, that's the 78th version. So it's beneficial for doctors and patients and gives back to my community. And as far as that project is concerned, that is all that there is for that. The other project I work on is, like I said, the ruthenium synthesis, and the design is to try and make a better photoelectric cell for solar cells. And we take a metal ion and we complex it with a purity ligand. And the goal is to make it as steric, which means as tight and condensed as possible, so it emits light. And that's more of an inorganic chemistry specialty. That's pretty much what I do.